Hello and welcome. We're back for another episode, and we're very much delighted. We have a guest today. Would you like to present our guest, Michael?
Absolutely, Markus. So, this is actually is it our third or fourth episode, Markus?
It should be the fourth one, I think.
Fourth, okay. So we are actually on a run. So we are continuing with our video presentation of what we do at Gabather. And as Markus said, today we have a guest, a very distinguished one, and on top of that. So the idea was to get somebody that Gabather works together with to interview, just ask them to see how they are seeing Gabather. And first idea here was actually to speak to Mahmoud Hassan. He is the CEO of Mindig. Mindig is a company that experts actually and specialists in EEG in particular, but also in imaging analysis. Mindig is a small company, it's based in France. Mahmoud himself is, I would say, top gun in EEG. And to tell you a little bit the background story, how I got to know Mahmoud and how we got to work together, and I think a very, very fruitful collaboration, is I was looking for somebody that could help us with the EEG data of our clinical trial. I thought that we had a lot of really good stuff, and I was trying to find somebody, and via internet, of course. I actually had a chat with Mahmoud. I called him up, and I said to Mahmoud, I think I have this and this ideas about EEG, and sometimes in life you meet somebody and it immediately works. We have the same idea, the same philosophy, and my idea was, can we use EEG for drug discovery? And Mahmoud wants to use EEG for, he'll explain later, for example, diagnosis, general development, not only from a scientific organization, but also if you look at the Mindig website, for example, they want to apply EEG for a purposeful, meaningful application, not beyond the interesting stuff with the EEG per se. So I got in touch with Mahmoud. He took a look at our first set of data on the EEG of our clinical trial of GT002 in the healthy volunteer studies that we performed in Portugal. Then Mahmoud took also a look at the second batch of the EEG data and the fMRI data together. So he's actually done most of the analysis of our stuff. I think we have presented some really interesting data. This is, and Mahmoud will probably, can explain more. This is not a trivial thing where we just press a button and you get the analysis out. This is actually handcrafted stuff and data that we got. And I think the value of the data that you get out of such a study, a clinical study, which was very important for Gabather, is directly related to the quality of the people that are actually doing the analysis to get it out. And I think we've been very lucky with Mahmoud. And I can also tell you that Mahmoud is also professor in Reykjavik, and he teaches there. And I've attended myself and all of Gabather to update us. I am not an EEG specialist. I'm a neuropharmacologist, an old-fashioned patch clamp guy. But we went to the summer school in Reykjavik. It was very excellent,
I think I saw a picture of that, actually.
Yeah, we had a picture with Mahmoud and me in the middle, right there, on the stairs at the university. Having said that, I will give the word to Mahmoud, just maybe to introduce himself very briefly. Then, Markus, if you have any questions, I will just have some follow-up or expansion thing. And maybe to the people that are listening, what Mahmoud is actually doing right now, and it's not only Mahmoud, he has also a team of very, very nice people in his group that do the analysis, of course. But right now we are actually working on getting the data together in a public, and eventually later on this year of the results from our study. And okay, I already talked much too long, Mahmoud. I give it to you. Maybe you can tell your part of the Gabather Mindig interaction story.
Yeah. Thank you so much, Michael. I think you already said it very well. So just to introduce myself very quickly. So I'm Mahmoud Hassan. I'm founder of Mindig. So Mindig is like for digging into the brain. This is the name came from. And we are a team of around 10 people here in Rennes, in the west of France. We are specialized in EEG. So Mindig created in September 2021. And the idea is because we believe a lot in the importance of the EEG, and not only in fundamental research, but also in translational research, and the EEG as a tool for, support in diagnosis, prediction, but also drug development. I'm also, an adjunct professor in Reykjavik University, and also recently co-founder of another company called Heila Labs. Hela is a brain in Icelandic, and also it's a company where we are combining AI with EEG for developing new tools for the assessment of brain aging, like normal and abnormal brain aging, and a tool for predicting some pathological neurodegenerative diseases. So yeah, this is very shortly what I'm doing. Yes, as Michael said, it was like a very nice coincidence when Michael called me. I was in Lausanne, I remember, and we discussed about the EEG data, and I was really so happy to see that the pharma industry and people in pharma industry are interested in including the EEG as a key part, not only for safety, which is like something traditional, I would say, for EEG in drug development, but also at next step. And this is, I think, the vision for later is to use EEG as also at some point as endpoint, in addition to the clinical assessment. Maybe we'll discuss about this later, but EEG has its place for, improving, I would say, the success of the clinical trials and drug development, not only in neurodegenerative diseases, but also in psychiatric. So it was really great to connect with Michael, and now we are working in that second project together, and I think third maybe soon. And I'm super happy to see, that Michael is pushing the use of the EEG in clinical trials, which is really, really great. I think this is the right way to improve, drug development in brain disorders in general.
Because this is quite novel also, right, Robin? I think we've talked about this before, but can't you two reiterate how are you usually doing it when you start assessing the candidates in patients and so on compared to what you're doing?
Well, EEG is more like generally used more as from a diagnostic approach, right?
Of course,
all-time classic EEG is, of course, studies in epilepsy. But I started with this way because one of the tricks with doing in a CNS program was, okay, how can you prove that your drug actually penetrates the brain and does change the activity? This is what we want to see. This is what we need to do if we want to progress this further into the clinic. And okay, you can have all sorts of biomarkers, but at the end of the day, the brain, and particularly in neuropsychiatry, where you're looking at behavior and emotions in the human brain, this is all done not at the level where I used to study it, which is in the old-fashioned test tube or the old-fashioned microscope here behind me in a patch clamping a cell. What you actually have to look at is the functional connectivity between areas of the brain talking to each other and generating perception, feelings, cognition, all this stuff. This is one level beyond me. That's why I needed somebody like Mahmoud that can actually study the EEG activity in the brain, not in the test tube. Not just saying that's not important and it's part of the drug development so far, but once you get into humans, and we knew that the drug was safe, we had to prove that this drug actually, this candidate actually does something also in the human brain. And the best way, the most elegant way, in my opinion, and I'm only being more convinced of that now than I was before, was to look at EEG. It's non-invasive, it's very, very fast, and it's progressing. And people like Mahmoud, they're really pioneering this, always one step forward. You heard AI is, of course, the next huge leap for EEG, I'm sure, from the number of electrodes, how deep you can go into the brain. This is all progressing all the time. But it was that activity of the brain that I wanted to see. In our case, not in a disease and a diagnostic approach, but in a drug. Does this drug do something? And with what Mahmoud already has found out, now I know a little bit, okay, something will happen in this area or that area of the brain. Okay, then I can go back and said, this could be interesting for this and that disease. Because remember, from Gabather, our molecules come from a department of chemistry at the bottom. These molecules were not designed to anything, but we tried to develop them. Are they potent? Are they safe? Are they selective? Okay. Then next step, how can we find out what's the best use of them? And in my opinion, it was EEG and then fMRI, which Hassan is also doing, Mahmoud is also studying now, was the best approach. I mean, Mahmoud, for example, it's a very interesting project which we are very happy to be part of. We're looking at EEG in a diagnostic setting with the Precise Consortium. It's super interesting, Mahmoud. If you can just do it.
What's that?
You put a colossal amount of work into this EIC. I think you should definitely mention it.
Yeah, exactly. And I think exactly in addition to what Michael said, I think the one of the big issue so far, I mean, maybe related to your question, Markus, EEG or neuroimaging in general, were used It's not yesterday, since many years. EEG exists since 100 years. It was
Of
the first
course.
EEG in 1924. And I mean, clinically used in epilepsy because it's easy to see epileptic seizure or epileptic spike on the EEG. No need for really very complicated, sophisticated analysis, which is not the case for psychiatry, neurodegenerative diseases, where really advanced analysis are needed to extract information and knowledge in biomarkers from EEG. But if we focus on drug development, one of the big issue is the one-size-fits-all problem that we have in drug development. It's like we take like a population of patients, let's say 100 patients, and we suppose that they are the same, they are coherent, they are homogeneous, which is not the case at all. There is a very high level of heterogeneity between the patients. And for this reason, all the effort that we are doing and also see the community is moving into is what we call precision psychiatry. And precision psychiatry, the idea is that treatment should be based on the individual biology rather than only the clinical presentation. So it's like based on the clinical, the biological profile. And then this is a key into like something at the patient level that can increase the success rate of the clinical trials. Because if we take like everybody, and I know there is no magic, medication that will work on all the patient with all the variability and complexity that we have between patients. So this is something very, very crucial. And to do this, data are key. So, I mean, we need data. And in terms of data, neuroimaging, so we have the MRI, so we have the structural MRI, but also we have the functional MRI, and also we have the EEG. Why we are like defending EEG more? For many reasons, I would say. EEG is non-invasive, so it's easy to use compared to other neuroimaging techniques. But there is also some key issues that we usually forget, which is EEG is scalable, which is, easily scalable compared to other neuroimaging techniques. It's also cost-effective. If you want really to spread this, solutions, it's impossible with the MRI, and also can be portable. We already have, at-home recordings now for EEG, which is impossible also with MRI. In addition to the excellent time resolution, our brain is very dynamic. It's not static. In EEG is a millisecond resolution, so we can see really how dynamics are changing, brain dynamics very, very fast. With all these, parameters and other parameters, EEG, I would say, is the best candidate to be used as a tool toward precision psychiatry or precision neurology in general, like improving drug development. And for this reason and many other reasons, like for example, what we call the reliability of the result, what we call the test-retest, how correlated your biomarker, at the instant T and T1 or T2, EEG showing a very, very high test-retest in very, different conditions, which is also very interesting for pushing EEG into a clinical use. So for all this reason, EEG, I would say, is a very, very good candidate. So I think defending EEG is not only for defending EEG, but also for a reason why, and even compared to some neuroimaging techniques. So mentioning the project that Michael talked about, so we are working now into developing, this precision, I would say precision brain health, that can be used in psychiatry, but also can be used in neurology. And the idea is to develop what we call, like the growth chart for height and weight for the kids. So developing the same for the brain function. So, we'll have like some reference, let's say from 0 to 100 for brain health, and then we can see the deviation at the patient level. So it's not like a group analysis. So we go beyond the average brain and then see at the patient level how deviated they are at specific age range. So we believe that this is really the next chapter if we succeed in this next chapter of the use of EEG as a clinical tool. And this tool can be used also in drug development because we can quantify scientifically the effect of a drug before and after, seeing how deviated from some normal trajectory and so on. So this is why EEG is key, and why in short term EEG will have its place in, as an endpoint in, I would say, short and midterm in drug development and global, I would say, brain assessment.
So you can see, if I can just append to what Mahmoud just said, that we had a situation where Gabather, we had a molecule quite well described. I mean, I've done a lot of preclinical stuff, non-EEG, and we knew where we wanted to get to. And Mahmoud was on the other side. I said, well, you can use this, and it would be nice to have something like GT002 to show we can actually pioneer this from the very, and if we succeed, then we will be able to document from the very beginning, I mean, literally from the chemistry of this molecule to the EEG and the other end, and looking at functional connectivity in a disease versus a control versus a healthy brain for the same molecule. And that is, classical, proof of principle. This is what you can do with this technique, apply to drug discovery with this molecule as proven by GT002. Okay, now I'm very optimistic, but I'm optimistic by default. But that is what the potential I can see in this project, and then Mahmoud also could see in this project. I think that is how this whole thing started, actually. And I think it's been absolutely great. And we're starting a trial now on the patients in schizophrenia, so we get the data also that, and then we'll be part of the normative approach that Mahmoud had talked about. And I think precision in psychiatry treatment, Mahmoud, and also Markus, is the one thing that has always been missing. And I assure you that, not so many years from now in the future, people would say, the kind of treatments we had in the 1950s and 60s, they were just barbaric. They're totally unprecise. It's like hitting somebody in the head with a hammer. But now you can have actual precision where you can make a profile, this pattern in this parameter by, for example, EEG, will necessitate this kind of drug because it will normalize or restore this kind of pattern. I mean, okay, now we're talking quite far into the future, but Mahmoud says only a couple of years. But that would be an idea, that would be a fantastic way forward, I think.
So if I get it right, then we had this previous study, and we did this conclusion based on EEG data, and I can see how glad Robin was when he received the data. What you're saying is you will have the same setup then when you do the study in Denmark. Is that what you're saying, Robin?
Yeah.
Collecting the data.
We will then collect the data then, again, based on EEG. And then we do an EEG pattern and a control substance and control and GT002, and then we will also have a cognition test, and that data then we will then also of course share with Mahmoud. He's the top expert in that. We will include this data in the EE Precise project with Mahmoud, because the thing is, if you want to do the kind of normative approach that Mahmoud has, you need data. You need to see, okay, this is where the people how the distribution will look like, because then you have something to compare an outlier or somebody that's in for a precision approach. You need this kind of stuff, and I think it's a great start. And really, the consortium that Mahmoud has got together is really very, very powerful. And I'm very happy that Gabather is there as an industrial partner. It's nice. It's very promising, I think. But this competitive grant, EIC grants, they are tough to get.
Can't you give me some more background on that grant that you're talking about? I don't know if everyone that's been listening to Gabather before knows about it.
Well, that I leave to Mahmoud. Can you talk about EIC Precise in 2 sentences?
EIC is like one of the European called EIC. It's there is what we call ERC, which is like for research grant. It's like more for researcher. EIC for innovation. So you have 3 kind of grant: EIC Pathfinder, which is like TRL 1 to 4. So that we're still in the idea, the prototype. Then you have accelerator and transition. So the idea is that you should have some breakthrough, but also in terms of research, but also you should have the product and the impact in the application in mind. So we have this console. It's submitted. So I hope the reviewers are listening to us and they will be convinced. And the answer will be like in a few months. But the idea is we have people who know how to analyze the data, people who know how to do the encryption and the security of the data. We have different psychiatric, clinical centers, and also we have Gabather for a use case on the drug development. So we have all the ingredients that should have to build the first brain growth chart that can be used in clinical application for neural development, but also in psychiatry and later for neurology. So this is the idea. We cannot say more now. I mean, we'll wait for the for the feedback at some point. Yeah.
Yeah. But it's so interesting following the company and also seeing the partnership with Mindig
Sure.
that, it's not only GT02 that's moving forward, it's also how you do research. And that's very exciting to hear, actually.
Sure. Well, maybe a word on this. Mindig is more or less a research lab in the private, I would say. And we
Okay.
are involved in many different research projects, going from normal functioning into clinical application, psychiatry, neurology. Our idea, my vision from the beginning when I created Mindig is really to be the EEG partner, because we know that a lot of people are interested in EEG, and they should, as a researcher, clinician, psychiatrist, but sometimes they are afraid about the EEG because it's be sure about the data quality, but mainly for extracting the right knowledge, the right biomarker. It's not a press button. It will be never a press button. It's like a know-how that it took time. And also, it's something should be really well thought. It's not something that we run just a code and we have a result. So for this reason, we are like a partner in many projects at the national, European, and international level in research. I mean, with researchers, also clinicians, but also industry, like the project we have with Gabather.
Say, when you receive that, it's a big amount of data. Is it a bit exploratory as well, or do you have a set way of start tackling all the data?
Do you mean the data from Gabather or like in
Exactly,
general?
yeah, in general or from Gabather as well, because as we say, like they are so sensitive, these measurements and so on. Is there a lot of cleaning of data and so on that you need to take into account?
Exactly. And this is one of the big issue in EEG where really expertise is needed is what we call the pre-processing. Is the data, they are usually noisy, and the data were a bit noisy because with simultaneous EEG fMRI, you have a lot of noise that can affect the EEG. And the first step before doing any analysis is to be sure that you can remove this artifact, artifact coming from the machine, coming from a muscle, coming from the eye blinks, and so on. So this is a key step. And in the team, we were working in the last 3 years that we built a very solid, validated pipeline, almost fully automatic for pre-processing EEG data with like some quality control metrics that we know at the end if we can keep this data or not. It also will depend on the condition of collecting the data because, if you have garbage in, you will have garbage out. Whatever you can do in the pre-processing, it will not help. So we should be also should be a need also for being sure that the data were collected in the good condition. This is something that we usually also are involved in this part, that we can help from the beginning before running the real data, doing some trial to be sure that the data are of high quality, because if it's not the case, it will be too late later to get, to extract information from the data.
Yeah, of course.
That shouldn't surprise you as a software engineer, Markus, anyway. I knew because what Mahmoud already hinted that. I remember that our data was fMRI and EEG simultaneously. So I already knew at the beginning that you need to have somebody, very much. This is not, I can only reiterate, this is not to press a button and then get the result out at the other end. And the more I've been, again, I'm neither an engineer nor am I an EEG expert, I'm just a pharmacologist, But the more I got into this, the more I knew that, we needed really some expert to handle this if we wanted to get any good analytical data out of this study at all for Gabather, So it is, in my opinion, this is somewhere at the borderline. It has become between science and art, You really have to have this feeling for this is an artifact, this is not an artifact. Nothing you can just program in. I think it's been very good, and the data that we have now, we're going to use. We build on the data then that we have in the study with patients, and there will be other studies that we can do based on the same EEG platform to be used in drug development. So the whole circle comes back to the very beginning, to the first phone call between Mahmoud and myself. If we can do something with this data in drug development and with EEG on it. And it's really nice. If it really works, and we really done some pioneering work. Again, you can track something from the very, very beginning of drug discovery to a clinical effect in EEG in patient to treat a disease. That's fantastic for me. I'm more than happy with something like that.
Have you had any highlights from the collaboration that you have had? Because it's been some time now that you have been collaborating. Anything that stands out in the amount of time that you have collaborated over?
Well, if I just start, Mahmoud, I think it's been, first of all, from a standpoint, it's very good that Mindig, they've been extremely engaged in what you do. So sometimes if you give a project away, said, can you do this? Everybody will say, yeah, I can do that. Everybody. But very few will actually do it, and even the fewer will do it in the amount of time that you wanted to. So that already from us is absolutely brilliant. I would put the highlight there at the very beginning. And the second thing was also, that what I found very good is people always ask me, oh, yeah, but when can we get the results? Why don't you have any results, And I felt very safe knowing that this will take the time it has to take, and I always defended it, because the more time you spend taking care with the analysis of the data, the better the quality of the data and the statements that you can make based on that. I think that is that. And I mean, look, if we went to the EG summer school in Reykjavik that Mahmoud organized, not for nothing, I think it's absolutely brilliant. And the icing on the cake was the last data that Mahmoud and his team and his collaborator Aya produced, was that we could see that there was some coherence between the fMRI data, which is brilliant, that in the same lab, Mahmoud's lab, that the EG data comes from, there was coherence between these two kind of data. It was two different approaches, of course, fMRI and EEG. You have spatial versus temporal resolution and so on and so on. So I think it's absolute brilliant. I see, and I agree with Mahmoud, I can see the way forward, hence about cost efficiency and, in translational sciences, EG, with AI, on top of it, will really, really be in the forefront for the next couple of years. And fMRI will always be very elegant and nice images, but it would still be the cost and the resources needed will limit it, and it's much more of an effort compared to in EG. As Mahmoud said, I mean, right on the internet you can buy your own EG kit to do at home to combat stress or whatever,
A bit of noise
data I don't want to comment upon, but I think that is what I would say. What do you think, Mahmoud? Isn't it roughly along those lines that we've been working along, I think?
Yeah, exactly. I totally agree. And I would add that EEG is not easy to analyze. It's a complex, signal and data to analyze. And don't believe of anybody saying they have the magic box and you click, something and you have output. It doesn't exist so far. Maybe one day we will have a foundation model, a ChatGPT of EEG, where we can, have the output very clever. We don't have yet the data nor the model. Maybe some people are working on this. I know some people working on this, but this will take a bit of time. So far, it needs to be like, it takes a time that it takes. And exactly as Michael said, is in research usually takes time. I was so happy to have one of the collaborators that I will really say thank to Aya Kabara. She is really expert in EEG, and it was really very exceptional time, a very short time providing all the results, but because she is expert like for more than 10 years, and everybody in the team also, they are expert in EEG for like at least 7 to 10 years. And this is what I was saying, this know-how is accumulation, of experience with time, and why we are trying to not selling also, the EEG as a magical tool, that it's still in the research part, still like a lot of things to deal with. Michael mentioned this summer school that we organized in Reykjavik, and that people came out at the conclusion that they have a lot of doubt about the EEG, which was really, this is what really wanted to push into. It's like to be very critical about the things and not just run things and believe in some gadget that you buy somewhere to see like a very beautiful signal. We are not there yet, and then we can see clearly why EEG is not used. Why EEG is not used as a clinical tool in psychiatric or Parkinson or dementia or Alzheimer. It's not yet, but it will come, I think, for sure at some point. But because it's still some research that should be done in terms of like dealing with the variability of the EEG, the complexity, what we call the analytical variability, is like how to choose your parameters and your methods, that you select. We already have something that is validated, robust, with test, retest, and so on. But I think there's still some work to do in this direction before that the EEG can become something really used in the daily, I would say, clinical use, yes. And I think we started with the work of the EEG together at the beginning, and then the fMRI data later. So maybe we can just mention that it was really super interesting to look at this data, which was, as I said, EEG is always noisy and hard to work with, but at the end, with all the expertise, we saw really a very interesting pattern in the GT002 compared to now what we compared with the placebo and orazepam, and mainly in some specific frequency band of the EEG, which is something also very special to EEG, not the case of other neuroimaging techniques, that you can look at brain waves. And mainly in the alpha band, we have some very nice, significant, increase of this alpha at this very specific time, in very specific also, brain areas. And this was quite interesting to see only in GT02 and not in the other conditions. That was something, quite interesting as a pattern that, in the next step can be, I think used in the next phase as maybe a biomarker to target, at the next step. And this, usually without like a lot of extrapolation and hacking is, this alpha wave is usually associated with the improve of relaxation and less anxiety and so on, which is like one of the main motivation for GT.
So when you listen to that, Robin, with the alpha waves and so on, what's your feeling? Do you think correlating that to Denmark, is there any hopes from your side?
Well, definitively. The thing is that, once you're talking about things like relaxation, mental states and so on, then you're really at the borderline approaching between what is still physiology and you're already going into either medicine or even further away into philosophy, because you're actually talking about mind processes, okay? So you're talking about what we will be able then to correlate with what was in Denmark. We've just put the foundation. These were healthy volunteers that Mahmoud and his team looked at together. We looked at whether EEG, fMRI, and we still have, a whole set of data where we had the same volunteers doing a task at the same time as the EEG was recorded. So now we can see, okay, how does the brain react when it's challenged with a certain cognitive task and numbers to remember and so on. And then we can compare the EEG and fMRI. That analysis has not been done. That will be the next step. It will come after the summer, and will be done, of course, also by Mindig as a next step. And as we started the study in Denmark, the data will eventually also be reviewed based on what we already found, compared directly with Mahmoud. So it's in that sense, it's really, everybody wins in this kind of interaction. We get insight into Gabather. The EEG as a technique and a diagnostic tool gets involved into this, and the patient maybe also have some eventual, if GT002 can show that this is a way of either biomarker or diagnostic, and also for us, we not only have GT002, there's also something called GT005, 4, and 6, Now we have all the reference, and I can assure you that if we ever get so far and getting the next compounds into a clinical candidate that can be done, the first thing we'll be looking at what EEG pattern does this compound have compared to 002. And remember, we have this battery of data on preclinical assays that we've done on GT002, which is huge, which is big. And then we can use all this, and then we can combine it with what Mahmoud is producing with Mindig and the EEG. And you have actually the whole package. You can follow it from the very beginning, from the very simplest biochemical assays to an EEG data. We're looking at alpha waves and relaxation states in the healthy brain. That's a fantastic story, in my opinion. But that is just, from a scientific point of view, it's very coherent. And that's why I think it's very exciting. You ask about exciting. So the story is not over yet at all. It's a EEG, fMRI. Now we're looking at tasks. We're publishing that. Mahmoud is already working on the publications of the data that we have so far. So there's a lot to do, and imagine that Mahmoud is successful with the EIC grant. That would be a fantastic thing. It's a huge network of really good groups that he got together, clinicians, EEG experts and so on for this, and having a Gabather there as a pharma partner is a fantastic story. I see this is not an asymmetric thing where one people gets a lot of benefit and the others don't. This is I think, if there's something called win-win, I would say it's a win-win-win-win-win. I don't know how many people we have in the consortium situation,
Sounds more like a partnership almost, right? Going hand in hand in this.
At the bottom line, that is because the ultimate goal benefits everybody. Mahmoud is as much appreciative of positive results as I am, of interesting results on the progress, I am as much interested in having a progress as Mahmoud is, we don't have there's no difference, and everybody in the consortium is, because the eventual goal is the same. And I come back to that very first phone call, which we found out when Mahmoud was in Lausanne drinking coffee, and he got a phone call from me, that is where I could see this whole thing getting there, where we can actually get within this year or next year latest.
You mentioned that there's going to be a conference or something in the future, Mahmoud. Are you going to present the EIC or something in a conference in the future, or how does that process work?
The EIC is submitted, so we wait for the answer in October. So we will receive the feedback in October. After that, we will start the project. And then once the project is started, then we can, present it everywhere. But so far, we can do nothing until we got the feedback.
Okay, sounds exciting.
Check out the Mindig website. They have all their publications and presentations there. There's very nice stuff on EG, and really top gun journals. It's really, if you want to know more about Mindig background, check the publication list on the website. It's very impressive.
I actually started looking through that list, and it's very impressive. Great work on that.
Maybe I can add something. I think something following this discussion about the EEG and the clinical trial. I think something that should mention what we already done with Michael on the first data is a first step of the importance of EEG in drug development, which is more in the safety, the effect of the new drug. But what we believe that the EEG has really place is at least, just mentioning two key applications. The first one is EEG to stratify patients to be included in clinical trials. It means that we use EEG even before starting that patient recruitment. So this is one of the big things that our team is working, is doing what we call, stratification is a word for finding subtyping, clustering, if you want a technical word, clusters within the patient, because the patient, they are very, very heterogeneous. And there is some subtype of patient that they maybe will respond differently to some drug compared to others. So looking at this stratification, finding, identification of the subtype of patients will probably increase, the signal to noise ratio and then, the efficiency of the developed drug. And this is something, in my opinion, one of the concrete application of the EEG, not only on the healthy subject for safety, but also to select, stratify patients to be included in the clinical trials based on their biological profile. So this is something very interesting, in my opinion, and some colleagues in different groups, they started to look into it for different applications, which is something I think very exciting. And the second thing is that EEG start to be tested as a tool to look at the responders to some therapy. It's like that EEG, because now what we do is trial and error. It's you try a therapy, you come after 1 month, whatever all the effect side, the side that you have is negative sides, and then okay, let's try another thing. And the idea is that EEG can be used to select the right therapy for people. I know some colleagues working on this in depression with really very exciting results, showing that for a specific patient, maybe if brain stimulation is working better than a medication, or maybe this kind of therapy is working better than the other, and then you can predict what therapy it's oriented the patient to the therapy that will work for their biological profile. So this is only the two application, in my opinion, that related to what we talked about drug development. Is beyond this traditional safety that we already started to do, but at the next step, this stratification and use EEG as endpoint, treatment that can be developed based on the biological profile, not only the questionnaire, which is and self-reports, as subjective as they are, as we know, The idea is not like to replace this clinical assessment by the EEG, but like maybe a combination of both can be interesting, but it's not enough. It's not enough. We need to look at the biological profile.
Very interesting talk today. I want to thank you both for attending. It was great to meet you, Mahmoud, and please check
Thanks
out
so much.
Mindig's website also. It's mindig.io. Check it out.
Yeah.
I think it's well worth a read. And until next time then.
Yeah, thank you so much.
Thank you.